Okay, this is a quick video to either revise or to study section 17.8, which is about epistasis. So epistasis is another example of a genetic cross. It's not a different way of drawing a genetic cross, it's just like a different result. So epistasis is when one gene doesn't code for a polypeptide, but it acts as like a molecular switch, so it switches another gene off. So it's a bit like drawing out a dihybrid cross because you're looking at two genes, gene A and gene B, but one of those genes will code for a protein and the other one just masks or modifies another gene's effects. You'll see what I mean in a sec when we look at an example. And the way you can kind of spot this happening in biology is if you get less variation than you would expect. So if this was, forgetting this for now, if this was a normal dihybrid cross, you might, from past experience, be able to work out what the ratio would be of offspring produced if you did this cross. Have a little think, see if you can remember. This, when you've got two heterozygotes crossed together with a dihybrid cross, you get this 9:3 to 3:1 ratio. It's quite a common one. So you would get 9 individuals with all dominants, 9 individuals with a mixture, 3 half-halves, another 3 half-halves, and 1 with all recessives. You can go back and have a look at your dihybrid notes if you're not sure what I mean. So you would get 4 different phenotypes in a ratio of 9:3 to 3:1. So that's this question here. But let's look at these individuals. We've got gene A and gene B. So gene B, this must be a bird, let's call it a budgie. Capital B codes for blue feathers. Little b codes for white feathers. In biology, white is often the blank canvas colour. If you don't produce a colour at all, you will look white quite often. However, if you have a capital A allele, that will block the B gene from working at all. If you have a little a allele, it doesn't block the B gene from working. So let's look at this individual here. They've got the capital B for blue feathers, so you would expect them to look blue, but they've got an A allele, and we know A blocks the B gene. So this individual here will look white, as will this one, because it's the same genotype. So let's cross these two parents together and we see what we get. So just like a normal dihybrid cross, let's put the gametes in here. So there are 4 different combinations of gametes that each of these parents can produce. It's a bit squished in this box, I'm afraid. So we could have, well, let's just pause actually, see if you can actually remember how to do the combinations of gametes like a dihybrid cross, and then unpause when you're ready and I'll show you. So you can have big A, big B, a little a, big B, big A, little b, or little and little, and the same for this side. Okay, now we're going to draw these out just like a regular dihybrid cross. So I'm going to have my big Punnett square. Okay, and I'm going to move my gametes to the side. And I'm going to fill this Punnett square in just with the genotypes for now. Okay, so I'll shut up while I'm doing this and you fill it in just like you would a normal dihybrid cross, just like this. Great, I'm done now, so if you need more time, just pause until you're finished. Okay, the next thing we're going to do then is we're going to fill in the genotypes. So if there's a capital B present, this bird will have blue feathers, unless there is a big A, because the big A will block that blue feathers from being produced. So, for example, this bird here should have blue feathers because it's got capital B, but it's got a big A which will block the B, so the bird will end up looking like it has white feathers. This example here, this bird has got a big B, so it should have blue feathers, but the big A blocks the B, so it will look white. So see if you can keep working along here. I'll do it silently, and then you can maybe just check when I get to the end that you've got all your colours filled in correctly. Okay, I'm finished now, so pause if you need some more time. So remember we said if this was a normal dihybrid cross, I would expect a 9:3 to 3:1 ratio of the ones with big A's and big B's in, and then a 3 of the little a big B people, and then a 3 of the big A little b people, and then 1 of these ones at the end. So I would expect 4 different phenotypes. Here I've only got 2 different phenotypes, white and blue. So I've got a ratio here of white to blue of 13 to 3. Or you can simplify that down to 4 1/3 to 1. So epistasis, remember, reduces variation in phenotypes. I've gone from 4 phenotypes in a dihybrid cross to just 2. Let's look at this example. It's the same idea. This time we've got some mice, and this species of mouse has genes for fur colour. So capital A codes for the colour of the fur, and B acts as a block. A bit like this one, but the other way around. So if a mouse has got a capital A for colour production, it produces melanin. Like if you think about the mouse's hair, like that, it produces the melanin, the colour, in bands, and so the appearance is like a brown colour, like this little guy here. I know it's black and white, but hopefully you can kind of see he might be brown. We call that agouti. So you don't need to know this word agouti, but it commonly comes up in exam questions, so just be aware, just so you don't get weirded out. It's kind of a brown colour. We use this term when we're talking about animal fur colours rather than brown quite a lot. If there's a little a, the melanin production is like equal through the whole hair shaft, so the mouse looks black. If there's a big B gene, it doesn't block this allele, or these alleles. If there's a little b blocker, it blocks any colour production at all. And so you'll get white. But look at that, it's recessive. So you actually need to have little b, little b in order to look white. If you've got one big B, it won't do any blocking. So you'll either have your black or brown mouse. Okay, so let's imagine here we've got some parents. Big A, big B, little b. And we're going to cross them. Let's say that we've got matching ones. I know it's exactly the same as up here. So your gametes are going to be exactly the same, aren't you? And your Punnett square is going to be exactly the same in terms of the genotypes. But it's good practice to keep drawing it out. The phenotypes will be different. Okay, so what is this mouse going to look like? Well, let's check the genes. It's got a capital A, so it should have brown fur. but it hasn't got two little B's. The dominant B is a not blocker. So this mouse here will look brown. It will look agouti. If it says agouti in the question, just call it agouti, not brown, but just, you know, just so you're aware. So this time I'm crossing these two mice together. So just like we did before, let's draw out the gametes. It's good practice. I know it's tedious. So you draw yourself your big Punnett square. I'm going to just be a little bit lazy now because I've got the one that I did in class this morning with the group. So pause this video until you've finished your filling in, and I'm going to flash up my completed example. Okay. Again, I got a bit lazy. I was writing ag instead of agouti. Let's see if I've got some. I don't think I've got any coloured pens. I've got a gold Sharpie. Should we use that for agouti? So these individuals here are all going to look agouti. And the reason for that is they have a capital A allele, which codes for agouti, and that allele is not being blocked by two little b's. Remember, you need two little b's to block. These have got, at most, they've got one little b. And then I have got some black mice. Oh, I think I've missed an agouti one out, haven't I? So why are these mice black? Genetically, why are they black? They need to have two little A alleles, and they need for that allele not to be blocked by two little b's. So two little A alleles, and then not being blocked by two little b's. Now these four here, each of them has two little b's, so that's the blocking allele. So it doesn't matter what the A's are at all, they're all going to be blocked. So they won't have any black or agouti coloration, they'll just look white. So here we would have a ratio of 9 agouti to 4 white to 3 black. Some people asked in the class this morning, what order do you put these in a ratio? Don't worry about that at all. Count your numbers up. The AQA questions you'll see when you do them next week. It's always obvious what it wants you to do. It will say how many or what fraction of the mice were agouti, what percentage of the mice were agouti. It really won't ask you to do ratios like that. So. Or if it does ask you to do a ratio, it'll tell you exactly. So like, let's say it says what is the ratio of white to black, then 4 to 3. So just look at the order of the words. Okay, so pause if you need to just get that sorted. And then we moved on and we looked at just this diagram here. So this diagram is what we call a biochemical pathway. Sometimes in exam questions it'll say, this diagram shows a biochemical pathway of some different chemicals being produced. So what this is showing is, like with fur colour, if you start out with a white chemical and that white chemical enters the active site of enzyme A, it will turn that substrate into a brown product. If that brown product goes in the active site of enzyme B will change the brown substrate into a black product. And we know enzymes are proteins, and we know proteins are coded for by genes. So because we've got two different enzymes, we need two different genes. So we just wanted to make sure people understood this diagram. So for instance, if there was a mutation in gene A, so that gene A no longer worked, what colour would the animal be? The animal would be white because there's no enzyme produced to convert the white substrate into the brown one. Even though this works perfectly fine, you're not going to do that step, and this step precedes that one. If there was a mutation in gene B, but gene A was absolutely fine, what colour would the mouse be? The mouse would be brown, because you can convert the white substrate into the brown product, but you can't convert the brown product into the black product. So a lot of these coloration changes are caused by different copies of these different genes. We don't need to do anything with that as long as you just understand how it works. These coloured chemicals here, like melanin was mentioned before, just check you know the word pigment. A pigment just means a coloured chemical. Alright, so just to finish off this lesson, we've done 2 examples. I don't really have many more examples to show you, but I am desperately trying to dig some out for this on autosomal linkages. These are the summary questions from the back of the book, from 17.8. So the answers are in the back of your textbook for you to check. They're not easy. They do take a bit of backtracking. So you'll get examples where you're told what the outcome is, but you have to work backwards and figure out what the parent is. So these did take quite a lot of deducing. People were very puzzled doing these in class, but we did get there in the end. But again, the answers are in the back of the book if you need some help. So please just upload the answers to these questions so I can see you've done them.